Hello Biology, today we are going to discuss chapter 8.3, and that is an overview of photosynthesis, the light dependent and light independent reactions. So to start us off, we need to take a look at our leaf, and we know that leaves are made out of cells. So leaf cells are going to be almost square because of that cell wall, and they're going to have tons of these organelles in them. And if you can remember those organelles, we would call our chloroplasts. And our chloroplasts then have structures in them that kind of look like this. So I call them, they almost look like sprees or individual sprees, and we call those thylakoids. And that's where our first reaction is going to take place across the thylakoid membrane of the chloroplast. So our light dependent reaction right here is going to take place in the thylakoid. So let's go ahead and label that. So our thylakoid membrane. And remember, that's just within the chloroplast. And our thylakoid membrane is going to be a lipid bilayer similar to the chloroplast in our cell membrane. So that's what's going to give it its overall shape, and similar to our cell membrane, we're also going to have important proteins in them and certain enzymes. So to start us off, we know that we need our light energy, and light energy is going to hit our pigments, and our pigments are found in our photosystems. So we have our photosystem 2, we call it PS2, and our photosystem 1. Now I know it's kind of confusing that photosystem 2 comes before photosystem 1. However, it was just discovered first, or I'm sorry, photosystem 1 was discovered first and then photosystem 2, so that's why it's coined those terms. So remember, the light dependent reaction always starts with photosystem 2. Alright, so what do we need for the light dependent reaction? Well, we obviously have our H2O here. So we need that. We also need then our light energy. And so I'm going to draw that in a squiggly form hitting our photosystems. So what this does is that the light energy is then going to excite electrons within our photosystem. So we have our electrons are excited right here. And so I'm going to do that via an E. And electrons are then picked up, and it also allows us to break apart water, okay, giving us a bunch of hydrogen ions. So we're using that light energy to break up that water, giving us the hydrogen ions, and then also producing oxygen, which is not kept in the membrane but is released into the atmosphere. Okay, so we have our hydrogen ions, and our light energy is going to do that for us, and we can make sure we do that. Light still there. And so what's going to happen is we're going to start transporting this electron through a series of events. Now, I'm not going to make you remember the specific names of these different proteins. Just realize that we have to transfer this electron, so we call it an electron transport chain. In order for us to transport electrons, what we do want to recognize is that even though we have tons of hydrogen ions here, we still have some hydrogen ions outside of our thylakoid. And what we want to do is we want to actively transport as many hydrogens as we can into the thylakoid space. And so transporting these electrons, we take some energy from that electron in order to transport those hydrogen ions into the thylakoid space. So as the electrons travel through the electron transport chain, we're also losing energy. So once we get to PS2, our electrons, we need to excite them again. Okay, so we need to go ahead and zap them with some more light energy. Okay, so we have our light energy, and as this is going on, we're having tons of hydrogen ions over here, and as you can see, we have a concentration gradient that's lower on this side, but it's higher up here. So this is one of those instances where we see active transport take place, where we're going from low concentration to high. And that's all for later for our production of ATP. So not only does this light then excite photosystem 1, but it also allows us to transfer NADP plus into NADPH, which is an electron carrier. So NADPH is going to pick up that electron, it's going to pick up a hydrogen ion, and it's going to then be used in the light-independent reaction. Alright, so once all of that's through, what's going to happen here is we're going to deal with our ATP synthase. So ATP synthase, we know that synthesis means to make, so let's go ahead and label this here. Okay, so ATP synthase, this is a protein pump for the most part, and what's going to happen is because we have a ton of hydrogen ions down here, they're going to want to flow from a high concentration to a low concentration, and they'll do that via ATP synthase. So think of a turbine and energy. ATP synthase is going to turn like a wheel, and because it's turning, what it's doing is it's harnessing energy, and as these hydrogen ions pump through, what that's allowing us to do is create ATP. Okay, so ATP from NADP plus a phosphate group. So all of that energy from the hydrogen ions going through ATP synthase allow us to collect ATP and NADPH, which are important for the next step in our reaction. Alright, so then what happens in our next reaction, our light-independent reaction? Well, we know that our light-independent reaction, we require 6 CO2. And we can also call this reaction the Calvin cycle. So, Calvin cycle. Because we are fixing carbons. Okay, so we are fixing carbons, and this is going to happen in the stroma of the chloroplast. So the stroma is the part of the chloroplast that's like the cytoplasm, so it's outside of the thylakoids. So before we can begin our Calvin cycle, the Calvin cycle is actually going to be a series of reactions that's cyclic. So it goes in cycles and things are reused. So the Calvin cycle actually starts off with already 6 5-carbon molecules. And then carbon dioxide is added, so CO2 is added to that. And when that's added to that, we break that up in step 3 into 12 free carbon molecules. Okay, well then we need to fix this even more. In order for us to fix this even more, we need to transfer some energy, and we need to transfer some hydrogen ions. So what we're going to do here is our ATP, and there's 12 ATP molecules, are going to transfer their energy, so then they turn into ADP, okay, so 12 ADP to be exact, to our 12 carbon molecules in order to create some bonds. And then we're also going to take the electron and the hydrogen ions from the NADPH, it's 12 NADPH, and that then transfers into NADP+. Okay, in order for us to get 12 molecules, so we have our 12 molecules of sugars that have a phosphate group, so that was given from the ATP. 1, 2, 3 carbons and hydrogens. Okay, so now we have 12 of those molecules. Well, 2 of those leave the cycle, so we have 2 molecules that look similar to the one before. They leave the cycle, and this is what's going to become the sugars, or whatever else is needed. So you can say our starch, our cellulose, whatever the plant needs. Okay, so that is used to make sugar. Well, then what about the other 10 molecules? The other 10 molecules are going to continue within the cycle. So they're going to travel this way. We have our 10 3-carbon sugar molecules. or 3 carbon molecules. We don't even have to call them sugar. Okay, and then we're going to require more ATP to then fix them into that 5 carbon group again. So we require specifically 6 ATP for this. So we have 6 ATP, and that is turned into AEP, which can then be used in our light dependent reaction. But then we go back to the beginning of our cycle, and this goes on and on and on and on. Okay, so that's photosynthesis broken down. It's a little less complex than the crash course video, but hopefully it helped you. Maybe watch this a few times to kind of put things into memory. But let me know if you have any questions.